Hello and welcome to Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. In this video, we will discuss about in vitro screening methods and their applications for natural products. Here, we will specifically have a look at the in vitro anti-inflammatory assay. Before going into the details about the assay, we will understand what is inflammation. It is the body's immune system's response to an intruder. Now here, the intruder can be anything like it can be a thorn, or it can be irritant, or it can even be a biological organism like bacteria, virus, or other organisms like worms. Sometimes the body mistakenly identifies its own cell or tissue as harmful, and this can lead to certain autoimmune diseases like arthritis or diabetes 1. There are numerous assay processes for determination of anti-inflammatory activity in vitro, like inhibition of the protein denaturation method or inhibition of 5-lipooxygenase, T cell proliferation assay, and the most commonly and most sensitive assay is COX enzyme inhibition assay. The COX means it is a cyclooxygenase enzyme. It is responsible for generation of prostanoids from arachidonic acid. Prostanoids means PGs like prostaglandin G2, prostaglandin H2, or prostacyclin, or even thromboxane, which are responsible for platelet aggregation. There are two isoforms of these COX, which are well known as COX-1 and COX-2. COX-1 is predominant in gastric mucosa and in kidney, whereas COX-2 generally elevates the level of is elevated during the inflammation process, or even pain, or due to bee stings or thorns, etc. Inhibition of the COX by these NSAIDs causes antipyretic effect, or anti-inflammatory effect, or analgesic effect. So that means these NSAIDs, they act by inhibiting these COX enzymes. Now we will discuss about the how COX helps in maintaining body activities, and why COX is to be inhibited. Suppose this is a cell. The cell membrane is made up of phospholipids, and there is an enzyme called phospholipase A2, which is secreted by the platelets. These phospholipase A2, they convert the phospholipids from the cell membrane into an arachidonic acid. Now this arachidonic acid forms the base for generation of PGs or prostaglandins. And how is this arachidonic acid? It acts by two ways. One by COX-1. The COX-1 enzyme, it converts this arachidonic acid to PGG2 or prostaglandin H2. Now, by this mechanism, if that means the COX-1, if it has converted arachidonic acid into PGG2 or PGH2, the same COX enzyme further takes these PGG2s and H2s towards the platelets and prevent or causes the aggregation of the platelet. It also protects the gastric mucosa and also maintains homeostasis, that is the balance of electrolytes in the kidney, or more specifically in the nephron, proximal convoluted tubule region. Suppose this is the normal physiology how platelets are aggregated during wound, or gastric mucosa protects the our GIs from acid attack or other toxins. But what if this enzyme, COX enzyme, is inhibited? Suppose if you consume low amount of aspirin or indomethacin, resveratrol or clopidogrel, what they do is they block this COX enzyme. Now, if COX enzyme is blocked, platelet aggregation stops. That means wound will not be healed fast at a faster rate or at normal rate. Further, gastric mucosa will not be generated. Instead, it will be eroded out of the body. And also it causes renal electrolyte imbalance. So these drugs, they inhibit the COX-1 enzyme. Or in other words, if you want to break down platelet aggregation, these drugs are prescribed. Another way is COX-2. If this arachidonic acid is converted by COX-2 enzyme to PGG2, the same products, that is PGG2 and PGH2, that results into generation of irritation or inflammation or even pain response. But these responses, they help an individual, or they alert an individual about the toxin that is entering into the body, or they indicate the organism about the threat from certain external or internal body parts, or say suppose foreign bodies. And if you want to relieve this pain or irritation or inflammation, COX-2 are to be blocked. COX-2s are blocked by heavy dose of aspirin, or normally all NSAIDs, they play an important role here. Further, coxibs like celecoxib, refecoxib and all, they also block the COX-2 and they prevent the inflammation, or they reduce the irritation or pain in an affected area or throughout the body. So this is how normally arachidonic acid forms the base for these activities, and either you can kill production of, or you can stop production of arachidonic acid by administration of certain corticosteroids, which specifically acts at the phospholipase, the first step. Here corticosteroids, they act and prevent formation of arachidonic acid. And most of the COX inhibitors, it can be COX-1 or COX-2, they act at these levels. Now, why we need to study this assay is because this is an in vitro method wherein COX enzyme is provided into the kit, then arachidonic acid is provided, and sample is also provided. So our samples or even inhibitors are also provided, and that means arachidonic acid, COX-1 and COX-2, then the inhibitors of COX-1, inhibitor of COX-2 are provided, plus we need to take the sample either from the tissue, or even standard sample is also provided. So if you want to know your isolated compound or extract or synthesized molecule, is it binding only COX-1, or that means selectively COX-1 inhibitor or selectively COX-2 inhibitor, or it is COX inhibitor in general, that means blocking both the enzymes. That means at synthetic chemistry lab, you can find hundreds of molecules generated by SAR mechanism, or by novel quantum as QSAR. Um, Various different chemicals can be generated simultaneously. Now, if you want to study the anti-inflammatory assay of 100 chemicals, it will take more time. So it is this in vitro assays, they help to perform the screening of all these at least 80 samples at a time in a single plate, that is a 96 well plate, wherein you can load these samples, along with that the load these reagents, and along with that our prepared samples are loaded in the same plate, and simultaneously you can get the result whether sample number 21 is more active or sample number 36 is more active. Accordingly you can synthesize, or accordingly you can isolate that molecule. So this is called as high throughput screening. The basic principle behind this method, or behind COX inhibition, actually there are various different mechanisms by which COX can be inhibited. The inhibition assay can be measured. The first is oxygen uptake, that means arachidonic acid. It is generally converted to PGG2 by this COX, and the oxygen that is taken up during this reaction is measured with the help of an oxygen electrode. Another method is oxidation of TMPD. TMPD is a chemical which upon oxidation gives a color, and the oxidation of this TMPD is done by the COX. COX is a dual enzyme. It oxidizes arachidonic acid to form PGG2, and further the same COX, it comes here and it causes peroxidation of PGG2 to PGH2. Now peroxidation of PGG2 to PGH2 is done in a test tube, and during this peroxidation it can also convert TMPD to oxidized TMPD. So now the oxidized TMPD has a color. This color is measured at 590 nanometers by colorimetry. Another method is by this finally endpoint assay. It forms a PGE2, and this is more accurate method. And here PGE2 ELISA reader is used to see how much amount of PGE2 is generated. So in this video, we will be discussing about the TMPD, oxidation of TMPD. This assay we will be considering. Background value. This is the blank for the value. To obtain reproducible results, the amount of cyclooxygenase, that is the enzyme, added to the well should fall within the range of the assay. It should not go beyond or below the range. The next, inhibitor well. Inhibitor means we have added the enzyme, we have added the samples, we are now we have also added the blanks. Now it is the time for us to add the inhibitors, the standard inhibitors. Assay buffer plus hemim plus sample is added, and to these samples the inhibitors are added. Now here DUP 697 is the standard COX-1 inhibitor, whereas SC 560 is the standard COX-2 inhibitor. So these two are serving as inhibitors, standard inhibitors, like our endomethacin or NSAID. Instead of adding an NSAID from the marketed tablet, the inhibitors are provided along with the kit, and these are to be added in 10 microliter proportion. So now here the inhibitor well will contain the assay buffer and hemim, and then sample, that is our sample. Suppose whatever we have isolated or prepared synthetically in the lab, along with that we are adding the standards, like either this standard or this, or you can add the combination, or that means few tubes should contain this and few should contain this along with the upper line, so as to know whether our sample, that is 40 microliter sample, is inhibiting SC560, that is COX inhibitor, COX2 enzyme is inhibiting, or whether you need to compare it with the SC560 inhibitor or DUP, that is COX1 inhibitor. Then the plates are shaken and incubated, and in the seventh step you need to add the major component, that is TMPD, which will develop the color. Now this TMPD is in the reduced form. The moment you add and incubate it further, it gets converted into oxidized version, and the oxidized TMPD gives the color or colored food complex which is to be measured. Now this is just an overview about the 96 well plates, 12 by 8, 96 wells. Now suppose this is the standard well, these are the two standard wells wherein you need to add only the COX enzyme. Then second, BS1 and BS2, background for sample 1 and sample 2. Suppose if you have only two samples to analyze, then in that case background should be there for both these samples. Then next, sample 1 and sample 2, whichever your samples, either in isolated form or synthetically prepared. These are the samples. Only samples are added in these wells and nothing else, along with the assay buffer and hemim. Then next, SC1 and SC2, that means COX inhibitor 1 in the is added, and the same enzyme is added to cross check whether so that the S2, that is sample 2, can be compared with SC2. These 6 wells will contain the assay buffer, hemim, and COX enzyme, sorry, COX1 inhibitor. Similarly, there is a COX2 inhibitor, that is DS1 and DS2, added in these wells. And lastly, the other samples, if you have range of samples, different samples, those can be added in these H wells. So suppose simultaneously you can analyze around, so suppose 9 into 8, that is 72 samples. Next is addition of arachidonic acid in these test tubes or in these wells, and needs to be incubated. Now you have the TMPD, you have the COX enzyme, you have the inhibitors, and now arachidonic acid, that is the substrate which will initiate the reaction. After incubation for 5 minutes at 25 degree, you need to record the absorbance at 590 nanometer with the plate reader, similar to the one which we have used in anti-cancer assays. Calculate the average absorbance. Now this average absorbance means sometimes we are using sample in triplicate, the background is also in triplicate, the certain COX are in duplicate. So first you need to take the averages and of the background sample and inhibitor treated sample, and subtract these backgrounds from their corresponding sample. That means the absorbance of background needs to be subtracted from the background sample. Only samples absorbance is to be subtracted from sample, and inhibitors from inhibitor. Now this, after subtraction, you will get some values. These are called as corrected absorbances or delta absorbances at 590 nanometer. Total COX activity, that means the enzyme activity for each sample, is the reaction rate at 590 nanometer, reaction rate of the enzyme, COX enzyme at 590 nanometer. And this can be determined by using TMPD's extinction coefficient, that is 0.00826 micromolar. 1 unit is defined as the amount of enzyme that will cause the oxidation of 1 nanomolar of TMPD per minute. The actual coefficient of TMPD has been adjusted for path length. Path length means very similar to our cuvette. In case of cuvette, we have 1 centimeters path length, and here you have 0.5 centimeter path length. Now the calculations, exact calculation is the corrected absorbance value for 5 minutes, and it is divided by the coefficient and multiplied by these factors 0.21 and 0.04 ml divided by 2, and this gives us the total COX activity per nanomolar per minute per ml, that is, or you can call it as units per ml. It takes two molecules of TMPD to reduce PgG2 to PgH2. Therefore, we are dividing it by 2. The total COX activity, subtract the total COX activity of each inhibitor treated sample from total COX activity of the corresponding sample. Suppose now here we have tubes wherein we have added the COX inhibitor and the sample, and also we have the activity of sample alone with the COX enzyme. That means COX enzyme, COX inhibitors, and the sample. The second well is COX enzyme and the sample. So you need to subtract the total COX activity of inhibitor treated sample from total COX activity of its corresponding sample. This is how we need to perform the assay, and the amount of inhibition corresponds to the amount either in COX1 or COX2 in the sample, or you can say both, whichever uh, enzyme you have used or whichever inhibitor you have used. Along with that, you have to compare. So this is how we perform COX enzyme inhibition assay. Although it is very lengthy process, but definitely it is very sensitive and most commonly used.